right, so hello. I'm recording this because just now when I conduct the class, okay, I forgot to record this part. Okay, so let me go make it full screen. Okay. Okay, let's wait, okay, I need to close my slide. All right, let's go full screen. All right, so now let's go back. I think this one is included in the previous video. Okay, now what is enzyme? Enzyme is an organic catalyst. Okay, catalyst means it will speed up a reaction. Okay, and mostly enzymes are made out of protein. Okay, protein. And of course, we have some add-on like carbohydrate and stuff. And the main function of enzyme is to speed up the biochemical reaction so that everything happens according to your body needs. You can see the green thing over here. Okay, it speed up the joining of these two molecules. Of course, it can join by itself, but with enzyme help, it can be faster. Okay, that is what the enzyme does. And let's talk about the necessity of an enzyme. Okay, why we need enzyme is because reaction does occur, but there are just too many reaction, too many biochemical reaction. Okay, and we need it to happen faster, so we need enzyme. Enzyme will speed up the biochemical reaction, and this will help us to maintain our life processes. Okay, where should I put this thing? I put down there. Yeah. Okay. Now next one. Okay. Now what are the how about the naming of the enzyme? Okay. Now enzyme have their own nomenclature, so they are on standard based on their own substrate. Substrate is what are the molecule that react with the enzyme. Okay. While reaction is the whole thing lah, the whole thing that is happening. So the enzyme name is usually based on the substrate, and you will see that enzyme always end with ASE. Okay. Just a few enzymes have different name because it was named before the systematic naming. Okay. So we have lactase, sucrase, maltase, and cellulase. All of this are an enzyme. Okay, now it was named after their substrate. Okay, we have lactose, sucrose, maltose, cellulose. So if I have a substrate called saccharase, okay, I might have a saccharase. Yeah, saccharase. I don't know. Okay. Okay, if a substrate called saccharine, maybe you got a protein called saccharine again. We have then an enzyme called saccharinase. Sounds good. Good name. Yeah. Okay, and now this is the reaction. The lactose plus water. This is hydrolysis. Okay, hydrolysis. We have glucose plus galactose. Okay, sucrose plus water, you're gonna get glucose plus fructose. Maltose plus water with the maltase, we're gonna have glucose plus glucose. Okay. All right, now this part is very important. Okay, very, 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 very super important. Okay, now we need to know that enzyme, it acts very rapidly. It is required in small amount. Okay, now this part I change a bit. Okay, it remain unchanged after the reaction. Okay, if you see the slide that I give you and the slide now is different. Okay, and it speeds up biochemical reaction faster, make it faster. And then it's very specific. So maltose is for maltase. Sucrase is for sucrose. You cannot change that. Okay, it's very specific. Okay, and then the reaction can be reversible, sometimes cannot. Okay, and also it can be inhibited or slowed down by what we call an enzyme inhibitor, like heavy metal. Okay, and some enzyme might need cofactor. That's why you need to take some protein like vitamin B or magnesium ion. Okay, all are, yeah, very important. Okay, next one is we're going to learn about how enzymes are made in your cell. We have intracellular cell. Intra means inside. Okay, while extra means outside. Okay, The intracellular enzyme is synthesized inside of the cell. Same is true with the extracellular enzyme. Okay, it's just that the intracellular enzyme stay in the cell and is being used by the own cell, while the extracellular enzyme will be secreted out of the cell. Now, example for intracellular enzyme is the hexokinase for the cellular respiration, while for extracellular enzyme is like the trypsin. So trypsin is something that being produced by your pancreatic cell, okay, but is secreted to the duodenum, okay, in the small intestine to break down polypeptide. Okay, so this will be on the, I think on the next part of the video. All right, that's all. Bye bye.